morning. The life of a plant begins from a tiny seed. How do seeds grow into a plant? The journey of a seed to a seedling passes through many stages of growth. Today, we are going to learn about the germination of seeds and the conditions required for the germination of seeds. How does a seed physically change during germination? And what are the conditions required for seeds to germinate? Last lesson, we have learned about fertilization. After fertilization, the ovary turns into the fruit. The ovule turns into the seed. The sepals, petals, stamen, star and stigma fall off. You have also learned about the structures of a seed and their functions. A seed is made up of an embryo coated with a testa or seed coat. The embryo is made up of a plumule, a radicle, and one or two cotyledons. Then it also has micropyle and hilum. Seeds are divided into two, which are monocotyledonous and dicotyledonous. What is germination? Germination is the growth of a seed into a seedling. Let's observe the physical changes during the germination process. First, the seed in the soil absorbs water through its micropyle and swells. Then, the testa splits open. The radicle comes out of the seed coat and grows downward. Next, the cotyledons spring out of the soil and the starch in the cotyledon is used for growth. Lateral roots grow out of the radicle so that more water can be absorbed. Plumule grows out of the cotyledon to produce new shoot. The shoots become the leaves. Once the first leaf appears, the seedling is able to make food for the plant. Finally, the cotyledons shrink and fall off, and the plant continues to grow. There are two types of germination, epigeal and hypogeal. For epigeal, cotyledons are carried out of the soil during germination. Examples of plants are mung bean and sunflower. For hypogeal, cotyledons remain inside the soil during germination. Examples of plants are corn, peas, and wheat. Look at the various types of seeds sold in the sundry shop. These seeds could have been stored for many years. Why don't the seeds germinate? Seeds need suitable environmental conditions for them to germinate. In order to determine the conditions, we need to carry out an experiment. 4 boiling tubes A, B, C, D are prepared as shown in the diagram. Boiling tube A contains wet cotton wool and some seeds, and it is placed at room temperature. Boiling tube B contains dry cotton wool and some seeds, and it is placed at room temperature too. For boiling tube C, some seeds are placed inside the boiling tube and some cold boiled water is poured inside, followed by a layer of oil. Cold boiled water is to remove all the air inside the water, and the layer of oil is to prevent the air from outside from entering into the water. Then it is also placed at the room temperature. For boiling tube D, wet cotton wool and some seeds are placed inside and the boiling tube is kept in refrigerator. All the 4 boiling tubes are wrapped with sugar paper. After a few days, these are the observations. For boiling tube A, it contains water, air, and suitable temperature. Therefore, the seeds germinate. For boiling tube B, it does not have water, but it contains air and suitable temperature. The seeds do not germinate. For boiling tube C, it has water and suitable temperature, but does not have air, and the seeds do not germinate. For boiling tube D, it has water and air, but it does not have suitable temperature, and the seeds do not germinate. So, boiling tube A acts as a control to show that if water, air, and suitable temperatures are present, then the seeds will germinate. All the 4 boiling tubes are wrapped with sugar paper to show that light is not required for germination. This is because during germination, the seeds obtain food from cotyledon or endosperm. Based on the experiment, you should arrive at this conclusion. In order to germinate, seed needs water, suitable temperature, and air or oxygen. So, water is needed to soften the testa and to activate the enzyme. The activated enzyme breaks down the stored starch in the cotyledon to glucose, which can be used in the respiration to supply energy for growth. Suitable temperature is required to activate the enzyme to function. Many seeds germinate at the temperature between 10 degrees Celsius to 35 degrees Celsius. Air or oxygen. Air contains oxygen, which is necessary for cell respiration. Oxygen enables seeds to respire to supply energy for growth. So, the seeds that are sown in the shop cannot germinate because the seeds do not have enough water, air, and suitable temperature. Next, let's answer a few questions. What are the steps involved in the physical changes in a seed during germination? So, step 1, testa splits. Step 2, radicle grows downwards. Step 3, lateral roots grow out of the radicle. Step 4, plumule grows out of the cotyledon. And step 5, the first leaves grow. Why the seeds in the test tube not germinate? This is because there is not enough air for the seeds to germinate. Which of the following factors are important for seed germination? Water, suitable temperature, and air. That's all for today's lesson. Thank you.